Hi all, I am Rosemary. I am going to take the seminar topic Plant Leaf Disease Detection. The first paper is Plant Leaf Detection and Disease Recognition using Deep Learning. Agriculture is one of the main factors that decide the growth of any country. Due to various seasonal conditions, the crop get infected by various kind of diseases. This disease firstly affect the leaves of the plant and later infected the whole plant. About how can identify the disease affected leaf by using CNN network. This proposed methodology it consists of three steps: image acquisition, image pre-processing, and then classification. First is image acquisition. Firstly, the images of the leaves were collected from plant villages and also some image dataset consists of 35,000 images divided into 32 different classes. The dataset which consists of both healthy and unhealthy leaves, which covers diseases like black rot, rust, bacterial spot, early blight, late blight, etc. Next step is image pre-processing. It consists of two steps, image resizing and noise removal. Image resizing means the images are resized into smaller pixel in order to speed up the computation. Noise removal means the accurate image contains some noise, so we have to remove the noise. So here we are using some techniques like Gaussian blur for removing the noise. Next is selection of the classifier. So here we are using convolutional neural network for the classification. CNN is far most complex deep learning model that we have used to classify the diseases. It is the most common neural network that is applied to image classification problems. CNN is a neural network which consists of four layers, namely convolutional layer, pooling layer, activation layer, and fully connected layer. It is the general architecture of CNN network, which consists of input, convolutional plus pooling layers, fully connected layer, and an output. First is convolutional layer. So here we are doing some mathematical operations to extract the features of the image. It uses some filters to find the feature map. So here the input image which consists of matrix of pixels, and here we are using 3 by 3 kernel. The kernel which slide over the input matrix and do some dot product, and then finally we get a feature map. Next is pooling layer. This layer reduces the size of the feature map and reduce the parameter numbers, training time, computation rate, and control over overfitting. So in figure, from first section, we are taking the maximum value 6, and from second section, we are taking the maximum value 7, and in third section, we are taking the maximum value 4, and in fourth section we are taking the maximum value 5. So here we are maximum pooling. That means taking the maximum value from each section to reduce the size of the feature map. Next is activation layer. In any neural network, activation layer plays an important role as it's responsible for nonlinear learning of the network. In our model, we have used ReLU for all layers except output layer. So here we are using activation function ReLU for all layer except output layer. ReLU means rectified linear unit, which is used for nonlinear operation. Next layer is fully connected layer. After performing all the computation in the previous layers, the output is fed into normal neural network for the classification purpose. Here we are using softmax max activation function for the classification in output layer. Next is training. The neural network is trained by using resized dataset, and we pass the trained data through the network and CNN layers which extract the required features and finally create a trained model. Here, the model trained 75 epochs using a batch of 32. Next is testing. Testing of the trained model is done by using test data. It is a graph. It's a graph of epoch versus accuracy. Blue line which shows the value loss, and the red line which shows the training loss. In starting section, the training loss and value loss are high. When epochs increases, the loss decreases, and we will get more accuracy. So higher epoch means higher accuracy in this case. So here we are using 75 epochs for the training. The comparison. The logistic regression which provides 66.4% accuracy, KN which provide 54.5% of accuracy, SVM which provide 53.4% of accuracy, and CNN which provide 98% of accuracy. So here CNN which provide 98% of accuracy, that means higher than any other methods. Next paper is plant disease detection using machine learning. This paper proposes the use of random forest to classify the diseased and healthy images. The dataset of the diseased and healthy leaves are collectively trained under random forest. For extracting the features, we are using histogram of an oriented gradient, that means HOG. Histogram of oriented gradient. HOG is an element descriptor utilized as a part of PC vision and image processing for the sake of object detection. Here we are making the utilization of three component descriptors. First is hue moments, then heraldic texture, and then color histogram. Hue moment is basically used to extract the shape of the leaves, and heraldic texture is used to get the texture of the leaves, and color histogram it is used to represent the distribution of colors in an image. First is hue moments. Hue moments help in describing the outline of a particular leaf. Hue moments are calculated over a single channel only. The first step involve converting RGB to grayscale, and then we are finding the hue moments. This step gives an array of shape descriptors. Next is heraldic texture. The healthy leaves and diseased leaves have different textures. We use heraldic texture feature to distinguish between the textures of healthy and diseased leaf. It is based on adjacency matrix which store the position of I, J. The texture is calculated based on frequency of pixel I occupying the position next to the pixel J. This is the general architecture of proposed model. First, we are doing HOG feature extraction, and then we are doing the training by using random forest. After training, we will get a RF model. For testing, first we are doing the HOG feature extraction of test image, and we are doing the classification by using RF model. Finally, we will get the result. Random forest. Random forest, which is used for the training of the model, and random forest which construct many decision trees during training, and decision of majority tree is taken as a final decision. So here we are taking the majority of decision as final decision. training. For each image in the labeled image, yes, first we are doing HOG feature extraction and add that to the training set. We are doing the training by using random forest and save the RF model. Next is testing. For testing, first we are doing HOG feature extraction of the test image. Then we load the RF model and classify the image by using diseases by using this model. And finally, we will get a result. Comparison with other methods. Logistic regression, which provides 65.33 percentage, support vector machine, which provide 40.33 percentage of accuracy, and k nearest neighbor, which provides 66.76 percentage of accuracy, and cart, which provides 64.66, random forest, which provide 70.14 percentage of accuracy, and neighbors, which provide 57.61. So here the random forest which provide higher accuracy than other models. Next paper is detection of potato disease using image segmentation and multi-class support vector machine. This paper proposed the use of multi-class support machine to classify the diseased and healthy images of the potato. In this work, we integrate the image processing and machine learning to allow diagnosing diseases from leaf images. The dataset, which consists of both healthy and unhealthy leaves, which covers the diseases like early blight, late blight of potato leaves. Dataset description. Dataset is collected from the plant village, and here we analyze 300 images of potato leaves and which have a spread of following class labels assigned to them. So here we are using three class label: late blight affected potato leaf, and early blight affected potato leaf, healthy or non-diseased potato leaf. These are the three classification labels. Next is training and testing. For training, first we are doing segmentation of the image, and then we are extracting the features from the image, and the class we create a classifier model by using SVM in support vector machine. Next is testing. For testing, we are doing segmentation of test image and doing some feature extraction from the test image, and classification done by using uh, classifier model, and finally we will get a result. Next is image segmentation. This is the first step. It segments the leaf from the background. We determine the threshold for L dash uh, A and B channel that will segmentize the leaf from the background. This task is performed by color threshold app in the MATLAB. Based on this threshold, we generate mask and remove the background. Next is feature extraction. Here we are extracting the 10 features from every leaf images of the dataset. The gray level co-occurrence matrix was used for extracting statistical texture features like contrast, correlation, energy, and homogeneity. Numerical indicators like mean, standard deviation, entropy, skew, and energy were calculated from histogram of color planes. Next is classification. Support vector machine is used for the classification of diseases. SVM is supervised learning model formally defined by separating hyperplane. So here we are using decision boundary in SCM for the classification purpose. comparison with the other models. Proposed method with the RGB imaging which provide 95% of accuracy. Hyperspectral imaging which provide 95 also 95% of accuracy, and texture analysis and TNN which provide 83% of accuracy, and K nearest neighbor which provide 82% of accuracy. So our proposed model which provide 95% of accuracy than other models. Thank you.